Welcome to the Catalysts podcast. I'm your host, Patrick Morley. Today, we have the privilege of speaking with the 2025 Taylor Prize laureate, Dr. Carl June from the University of Pennsylvania. For more than 3 decades, Dr. June has devoted his career to one big question: can we harness the immune system to cure cancer? What began as a bold and unlikely idea in the 1990s has become his life's work. The creation of CAR T-cell therapy, a treatment that has already saved thousands of lives. Today, we explore his journey, the persistence behind his discoveries, and how his lifelong pursuit of science is bringing us closer to cures once thought impossible. Thank you, Dr. June, for being here today. It's a sincere privilege.
Well, thank you, Patrick. It's a real honor to be on Catalyst. And I want to thank you, for the public service this does in letting the public know about these amazing advances in science.
Great. Wonderful. Yeah, we're very honored to have you here today for sure. We have a lot to talk about today, but first, can you tell me what happened on August 11th, 2011?
Well, sure. That summer was, a turning point in my life. Up until then, I was a scientist and physician working on, an idea for an immunotherapy. But in August, it became real. And it started off in an unlikely way with a patient at the end stage of his life for leukemia. And he volunteered for our CAR T-cell trial, which was taking blood from him, modifying it with viruses so genetically it could retarget, in theory at least, his cancer.
Yeah.
And we infused him August 1st, and then, he got really ill. And in fact, he had last rites done. He'd paid for his funeral. We all thought he was going to die. And then a bone marrow biopsy came back, and there was no cancer there. He ended up living through COVID and more than 10 years, never had his leukemia come back. It was one of these times where an idea, and it just, where this abrupt turning point, and you knew something that seemed impossible could be real.
Can you describe, first of all, that's an incredible story. I mean, you're given last rites, you're planning your funeral. I imagine his family was gathered around. And then all of a sudden this miraculous, it's not an exaggeration to call it miraculous, cure. He's cancer-free. Can you describe your feeling? Was it a colleague who called you to tell you, or was it through an email? Or
Sure. So, because my lab had made these cells that called CAR cells, what I did was at the back end make the cells, but then there was a physician named David Porter, who's a professor here and long-term colleague of mine, who took care of him on a day-to-day basis. And, David called me to say that, this biopsy came back and it said no evidence of leukemia. And I said, with a lot of skepticism, sure. What do you think that means? Do you think most likely they missed it? And how about we do another biopsy? Because that was on day 28 after treatment. And I was so skeptical. I thought it was impossible that it could come back that way, and they must have just missed it. And so they did it again. And on day 31, the same thing came back, this time from the other side. When you do a bone marrow biopsy for leukemia in adults, it's usually done in the hips.
Okay.
And so they went from one side to the other, and both sides came back. The official report was no leukemia, complete remission.
Amazing. Do you remember, or were you there, or did you get reports of that patient's reaction to that news?
Yeah. Well, I wasn't there. I mean, because David told him.
Yeah.
And his wife is Darla. The patient, has been on the New York Times. He came out with his name. It was Bill Ludwig. I mean, all that's been cleared because he authorized that.
Yeah.
It was an amazing event. He had a lot of grandchildren, and he was so grateful. And, he'd had a lot of courage to be the first person to volunteer to do this in the world.
Right. Yeah.
And, we were very grateful for that because we had no idea. And I mean, it turns out that it actually worked better in him and in our other patients than it ever did in the mice that we used to work this out. It was one of these a twist of fate where, because usually things work better in the animals, and then you try them in humans and you get this slap in the face and it doesn't work. That's what happens 99% of the time. But in this case, it had worked in mice, but not nearly as effectively as it ever has in humans.
Amazing. So I think the heart of our talk today, and one of many reasons you're receiving, the Taylor Prize is for your work and the CAR T cell therapy. And the big question is, the question I'd imagine you had obsessing over was, can our immune system be used and trained to cure cancer? And the definitive answer is yes. Can you walk me through how that process happens?
Yeah, it's actually a centuries-old debate, with the first idea that actually the immune system controlled infectious disease. And that happened in the 1700s when nursemaids noticed that people who got an infection from cows, by milking cows, they got a pox virus. And then they found out they were resistant to chicken pox and to smallpox, which was often lethal. And so then they started making vaccines from that. So that was the first intentional use of the immune system to change a disease. And in the 1850s in Germany, there was a pathologist named Paul Ehrlich. And they didn't know anything about the immune system, but he saw in tumors that they had taken out of patients under a microscope, because those had just been invented, that they could see these small round cells. And he envisioned maybe that those were actually fighting and making what he called magic bullets, to kill the tumor. And so that was before we knew anything about B and T cells. And then there were many efforts in the 1920s and 30s to augment the immune system by actually intentionally giving people bad infections at the time of a tumor diagnosis. And sometimes, these infections, what they did was take a bacteria and then basically boil it and take that extract out. They called that Coley's toxins. They would inject those into people. They got very high fevers. But once in a while, they actually had amazing regressions of the cancers. And that was the first really intentional try to harvest the immune system for cancer.
And how many years ago?
Got FDA approval.
Right. How many years ago was that?
So that was in the 1920s and 30s.
The 20s and 30s. Okay.
Yeah. The surgeon he started actually in the 1890s.
Right.
And he did it for many years in New York, and then his daughter carried it on. But they never could get a randomized trial for FDA approval. But it did show that sometimes that happens. But there was also toxicity. They started before there were antibiotics available. And so sometimes these infections got out of control and, there was danger to it. So it was actually then it fell by the wayside. And then what happened was in the 50s and 60s, radiation therapy and cancer chemotherapeutics were developed. And those became, other than what had been done for a very long time, surgery, the pillars of how we treat cancer. So surgery and then radiation and chemotherapy. And all the immune therapies had failed. So it was an old idea. And then, we wanted to see if we could, retarget the immune system from, the T cells that we have in our body. The public really learned about them during the AIDS epidemic, where if you lost your T cells, you started getting all kinds of unusual infections and actually died with a collapsed immune system. So
right, and T cells are they're the soldiers that attack and kill viruses, right?
Yeah, and so T cells are in our bodies, and you train them as you get older. As a child, you have a lot of T cells, but they're not experienced. And then you start getting vaccines and all the childhood infections, and then they give you lifelong protection against all the viruses and things that you get when you start going into childcare. And so that's what T cells can last the rest of your life. And so we have some of them that are dedicated against chickenpox, some against other viruses like COVID now. When you get a COVID vaccine, you're making both B and T cells make an immune response that protect you from, an infection. So T cells are really specialized for viruses. And our idea was maybe we could retarget them so that they could work also like a B cell. So B cells make antibodies. And during COVID, we learned a lot about antibodies against, for instance, the spike protein, which is on the COVID virus. And if you make these antibodies, that can protect you. So we made a hybrid of a B and a T cell.
Okay.
Where the T cell is endowed with a way to kill viral cells, and the antibody is made by B cells. And that, we used the Greek word for chimera, which is, a species made out of, it's a, an animal supposedly made of a goat and a serpent and a lion.
Right.
And so
a hybrid, so to speak.
Yeah, it's a hybrid or a chimeric species, and that mythical creature then was called a chimera. And so we made basically a mythical T cell and B cell, so that it's a fusion of the two. And so that's why they're called chimeric antigen receptor T cells, CAR T.
Right. Yeah. Do you doing the research for our talk today, I think there was a moment when you in the lab you made the CAR T, right? Again, that must have been a profound feeling, right? Can you explain that?
Yeah, I mean, to make one, as I mentioned, you have to get the gene, the DNA sequences that make an antibody, and transfer those and insert them into the T cell, so that the T cell now it's genetically modified. It now carries some DNA that'll instruct it, to make an antibody. And other labs like Michelle Sadelin's and New York's Memorial Sloan Kettering had worked on using a mouse virus to do that, called a gamma retrovirus. And we used actually the HIV virus because instead of being a mouse-derived virus, it actually is, came in humans, caused the HIV epidemic. But other labs had actually turned that deadly virus, the HIV virus, into a tool. And what they had done was there's about 10,000 base pairs in the HIV virus, and they cut out the ones that cause disease, but left the ones that lets it insert new genes into other cells. And in this case, we wanted to insert it into the patient's own T cells. So we used that HIV virus that was modified to insert the antibody DNA sequences permanently into the patient's T cells. And the first time it worked when we made humanized mice. And then those mice don't have an immune system. They're immune deficient. So what we do is then put the human immune system and install that into the mouse. And then we could give it tumors. And then, and then an infusion of our CAR T cells and ask what happens. And what we found was that the CAR T cells could really rapidly eliminate tumors. And then the most striking thing back in about 2008, 2009 was one of my postdocs, Carmine Carpenito, who's actually a Canadian, by the way. He showed me a lab printout from the experiment he'd done, and a mouse that he had forgotten to sacrifice. So every day we have to pay hotel rent on the mice in the labs. Basically that's called the per diem, to take care of them and to feed them. And, we had shown that the mouse got cured of cancer, but then the mouse was just hanging around in the cage. And Carmine decided to go analyze that mouse. And lo and behold, that mouse, 6 months after we treated him, was still chock full of these CAR T cells. And we could re-isolate them from the mouse and then show them in petri dishes that they could still kill leukemia cells.
Amazing.
So to me, that was a big aha moment that it's possible to retarget a cell, and it can continue to survive and long term, in this case, 6 months in a mouse like that after having eradicated the tumor. So
Right.
that was the decision where we said, wow, we really need to get this into humans.
Amazing. Now, this is a great example of science, right, at its purest form. And I think you've been kind of quoted, or I don't know if it's a direct quote, but you said something to the effect of, the art of science is knowing when to dig deep on unexpected findings. And I'm wondering if that comes from experience, intuition. How do you know, and how do you get your team to know, okay, this is the thing that we need to focus our energies on?
Yeah, it's so true. A lot of science is confirmatory. I like to use a basketball analogy. It's like getting a layup, and you get a 2-pointer, you should always make that.
Right.
But the big things in science are the 3-pointers, where you make a long shot, and oftentimes it misses, but when it does work, you get more points and
Right.
you learn a lot more. So in science, we do a lot of things where you have an expected answer, and it kind of confirms the models that we have. But sometimes you get an answer completely opposite of what you expected. And the easiest thing is to say, oh, that's, not to like that fact and just go on. But the best thing oftentimes is to go say, why is that? Because it challenges your core beliefs. And so sometimes it's, in fact, most likely is it was a bad experiment. You made a mistake. And when you go back and reproduce it, it comes out and you get the experiment, the result you thought you would get. But other times, when you find that, then you need to say, well, we got to dig into this and really see what's going on here. And we had several of those along the journey of making car cells. And one of them being, why did they survive long-term like that? I mean, I thought they'd be like short-term little killer bees, but they wouldn't live a long time like a queen bee and live, essentially for the lifetime of a mouse after we put them back in.
Right. Maybe this is a good time then to kind of introduce the Emily Whitehead case, which, if listeners and viewers haven't heard of, there's an incredible documentary, I believe from what year is that? 2022, I believe.
Yeah. That sounds right.
Called Of Medicine and Miracles. And it's was made by Academy Award-winning director Ross Kaufman about Emily Whitehead, who was cured using CAR T therapy of a pretty aggressive leukemia. One thing that's fascinating, I know, about her story is that all these years later, there are still CAR T cells active in her body. Did that surprise you?
Yeah, we all of the data we had been that when you make a cell that you genetically modify, it's likely to get, when you put it back into a human, a couple of things could happen, and all of them would be bad. Either it would die, it would kill maybe a cancer cell, but then it wouldn't survive, or it would actually get recognized by our own immune system as now being modified and rejected, because that's our immune system's job, is to look for changes there and let things that are of your own body stay there. But anything new, like a virus, bacteria, or like if you get a heart transplant, it's very hard not to have it rejected, even though it's a heart that came from someone else. The immune system will, unless you really give very potent medicines, it will reject it. And we actually thought that's what would happen with Emily. We wrote in our protocol that, we're going to give you these cells, about 100 million of them, and, they're your own cells, and we're going to make, this bullet in there that will be an antibody to retarget into hopefully your cancer leukemia cells, but that likely they will not live more than a month in your body because of this antibody that's in there is going to lead them to be rejected. And so that's what we thought would happen. That was in our consent documents. And, Emily was treated in 2012. And the way, both in Canada and in the US, when you start a new therapy, you first have to start in adults. And so we had actually developed the therapy that we made with CAR T cells on pediatric leukemia patient samples that my colleague Steve Grupp here at Children's Hospital in Philadelphia had a bank of them in the freezer. And so we would use those to see, as I mentioned earlier, put them into mice, and then the mice would get leukemia, and then we could use them to test if the CAR cells worked. And as I mentioned, they did around 2009. Then in 2011, then, we had actually treated Bill Ludwig.
Yeah, Bill.
And as the first and we treated 3 adults then. And then Emily Whitehead was the first pediatric patient to get treated. She had her cancer come back 3 times. She'd had all the best therapy you can get, followed all the rules, had hundreds of chemotherapy infusions and all the things that go with that. And so it was a last-ditch effort when we treated her. She had, when we treated her then, she had a violent reaction. We treated her on a Tuesday. In 2012. And by Friday, she had multi-organ failure with very high fevers, 106 degrees for 3 different days between Tuesday and Friday. And, the usual cause of a fever in a patient with an impaired immune system after chemotherapy, and they have leukemia, is infection.
Right.
But we could not find an infection. And so we looked, and what we found out was her immune system was hyperactivated. She had in the blood these cytokines. These are proteins made by the immune system actually to cause fever. So when you get an infection, it's not, for instance, you get the flu, it's not the actual virus that causes the fever. It's the fact that the virus stimulates some cells in your immune system. They put up a white flag, and they make these proteins that say we need a fever to help you fight the infection. And that's what was happening to Emily. She had one of those flags called interleukin-6 that was elevated over 1,000 times above her baseline. I mean, it was not even a question that was probably what was causing her 106-degree fever. And, she was comatose and had multiple organ failure. So it was really touch and go. The intensive care doctors thought that she was going to die. But it turned out there was a therapy that can treat the high levels of the interleukin-6, and it was actually developed for arthritis and autoimmune diseases, which is where your immune system is overactive. And by a lot of luck, it turned out my daughter had arthritis, and I knew about this drug for arthritis, and it had never been used in a cancer patient. But, we had our backs were against the wall with Emily. She had very low blood pressure and was thought to only have hours to live. And so we asked her parents, actually, she was comatose, but her parents and then our institutional review board, could we use this arthritis medicine called tocilizumab? Could we use that in Emily as an attempt to treat her high fevers and reaction to the CAR cells? And we got a very rapid approval, and she was treated on that Friday night. So that was 3.5 days after we had treated her. And then by Saturday, it was miraculous. She had no more fever.
It's such a story. Yeah, incredible, Dr. June. I mean, this is a story. There's so many little micro coincidences that happened to save Emily's life, right? I mean, the fact that you had that drug at your service is one of them, isn't it?
Yeah, I mean, it was in the formulary, and it had happened that in 2009 to 2010, I was president of the Clinical Immunology Society, which is a group of physicians and scientists that treat autoimmune disease, infections, and cancer. And every year the president of that society gets to give an award, and I gave it that year to Professor Tada Kishimoto, who's in Osaka, Japan. He had invented that drug. And, so he came to the U.S. and we had this award ceremony in 2010. And but no one had ever thought of using it for cancer patients. It got approved, by a pharmaceutical company, Roche, for arthritis.
Right.
And so it was sitting there in our pharmacy, and we were staring at Emily with this problem of very high levels of the cytokine that drug was approved for.
Yeah.
And now, so it worked amazingly well, and then we started using it every time.
Right.
And when it finally came to FDA approval in 2017 of those CAR T cells for leukemia in patients like Emily, for the first time, the FDA approved a drug, which was CAR T cells, and co-labeled and approved another drug with it, which is tocilizumab. So it's like a package. In
Right.
one way, you give the CAR T cells, but then if the patient starts to have fever, you give them the drug tocilizumab to block the fever. And so it really has probably saved that. I mean, because if Emily had died because of this very bad reaction she had, we would have never been able to go forward with a therapy. But it turns out it's very effective. Emily is now a junior at the University of Pennsylvania, 22 years old. And the most astonishing thing is to me, I mean, besides she's a young woman who's vibrant, studying English, and has no leukemia, but she still has CAR T cells now.
Incredible. Yeah.
Since 2012, it turned out to be a living drug, and
Right.
we never thought that could happen.
That's amazing. Yeah, I really do recommend, anyone. I actually downloaded or I rented the film on YouTube. So I think it was only $4, but I do recommend anyone who's interested in this to watch that film, because it really is Dr. Juna's story, not just of Emily. It's a journey of you, but and correct me if I'm wrong. It seems like the more I learned about you and your story to where you are today, so much of your life has sort of woven in and out of pretty major events of the 20th century. I mean, you were you entered the Naval Academy at the dawn of the Vietnam War. You've been through the turmoil of the 60s. Then, the AIDS epidemic of the 80s kind of led to the CAR T therapy. Then you have, I know, the Cold War sort of influenced the research into radiation, and then the wall fell, and you had, you needed to use all this information for something, and you used your experience in your brain to do that. Not to mention, the international collaboration or your knowledge of Dr. Kishimoto, right? That saved Emily's life. Is that fair to say that your, career and your life culminating, with the Taylor Prize, is sort of a story of the 20th century?
Yeah, I think, very much, Patrick. It's been unexpected. You cannot, a message to young listeners is you can't plan your career or your life and think, this is how it's going to be. And so for me, there's been, a lot of these turns in the road. I always like to use the quote from the baseball Hall of Fame catcher named Yogi Berra, and he used to say, if you come to a fork in the road, take it. And,
right.
That happened to me many times. I mean, the first time was in high school when I thought I was going to Stanford University, and then we were in the midst of the Vietnam War, and I had a low, the way they found out who would go into the Army or Navy, to fight in that war was based on a random lottery of what your birthday was.
Yeah.
And mine turned out to have a birth date that meant I was going into the military. And so I ended up, at the Naval Academy. And then I actually went to Vietnam when I was 19 years old. We took a bunch of Marines there on a ship. But the war got over when I graduated in 1970. When I graduated in 1975, the war was over and ended in 1974. So then the Navy let me train to be a physician, and because they needed people who could take care of bone marrow or radiation disasters. So, and that was because of the possibility of nuclear reactors having limited nuclear casualties. So in the case of the Navy, the worry was we had nuclear submarines going around following the Russians, but they're full of radiation, uranium, in a reactor. And if that could have an accident, then you would have people where the bone marrow transplant would be the only way to save them. So I was trained, and 4 people the Navy trained as physicians to be able to treat if that ever did happen. Fortunately, it never did to the U.S. I mean, we had events like Chernobyl in Russia. We had Fukushima in Japan, where reactors that were used for civilian nuclear power had accidents and caused severe casualties. But it never happened to the Navy's nuclear submarines, which is testimony, I think, to the engineering that they did.
Right,
So then what happened was in 1989, I was supposed to have a bone marrow transplant unit in the military hospital in Bethesda, Maryland, to treat these potential casualties. And then, in 1989, the Berlin Wall came down, Soviet communism collapsed, and there was no longer the threat of that nuclear war. So the Navy then let me begin just to work on the immune system, and rather than the nuclear casualty aspects. And that's when at that time, the big epidemic was HIV. And the Navy was unable, I'd been trained to be an oncologist for leukemia. That's when I was doing the radiation studies. But the Navy is not allowed to fund cancer research. They only fund through the Department of Defense research into infectious diseases like malaria, and that was my initial publications, and then, or HIV. And so I actually worked on HIV vaccines for about 5 years and on the HIV virus and how it impacted the immune system. And that turned out to be very fortuitous. I mean, it was one of these fork in the road. I had never even heard of an HIV virus, and then, but that was the way I could continue to work on the immune system, which is where my fascination was. And then, I got out of the Navy and moved to the University of Pennsylvania in 1999. And then I was able to get research funding for cancer, and in addition to our HIV. But the first CAR T cells we gave, I mean, a lot of people don't know this, was for patients who had AIDS. And it was to rebuild their immune system. It was only later, so we started those in the 1990s, late 1990s, and they were successful. But then drugs came out for HIV that have been very potent. And as long as you have access to the drugs, mostly it's a well-controlled infection. And so then, I began to apply what I had learned on HIV into cancer patients, and that led, finally to the first patients being treated in 2010 with results in 2011.
Amazing.
This mini-series is in support of the J. Allen Taylor International Prize in Medicine.
Correct me if I'm wrong, but I believe you were the first or one of the first labs to deal and handle patients with HIV and HIV blood. Is that correct?
Well, we were the first to grow. I mean, you had to have, it's called biosafety level 2 because the blood's infectious. And there were fears about that. It turns out actually now human blood is more contagious with hepatitis.
Right.
If a patient has hepatitis than it is HIV, but especially if they're under treatment. We didn't know that back then. And so we were especially worried because we were growing up large volumes of T cells that had HIV infection in them from the patients. And if we had, for instance, a centrifuge accident, that could make aerosols and potentially, contaminate us. So there, we had to dress up in bunny suits to protect us. But then we were able to show that we could grow large amounts of T cells from patients with very advanced AIDS, and then, reconstitute their immune system with infusion of their own cells that we had expanded. So we learned a lot from those experiences in an infectious disease that we later could apply to the leukemia patients.
And I think it's really important for listeners who, maybe weren't alive at that time just to understand just how there was so much unknown qualities about AIDS and HIV, and the bravery, I think, and courage it took for you and your team to deal with it and to do it, I think, is something that I want to emphasize and almost thank you for, in a sense.
Well, thank you, Patrick. I mean, it was, I think now people who've just lived through the COVID epidemic, we didn't initially know how it was caused, what the virus was, and then would there be a treatment for it? It was really bad, especially for older people or those who didn't have a healthy immune system. And back then with AIDS, we didn't initially know what caused it. And there was no therapy. Basically, it was a death sentence. So a lot of the people who got HIV early on in the 1980s basically sold their life insurance policies and just they knew they were walking dead.
Yeah.
And then basically the medical system did an amazing thing. They developed very potent drugs called protease inhibitors in 1998 or so. And that really changed that. It made it go from a always lethal disease, and it became then, if you had access to care, a manageable disease where people could have normal lives. So it was, a demonstration back then to that generation of how a new viral epidemic can really change everything. And it caused all kinds of fear in people because they didn't know would you catch it by walking into someone or whatever. And then we all went through that, a few years ago with COVID.
Right.
Going to the store, could you touch things in a supermarket? Would you get infected that way? So it really impacted society back then. And we have to really give thanks to the people who volunteered, both the scientists and the patients who suffered from that. Because in the end, medical science ended up, making a huge difference.
No, it's a wonderful story. To bring us back maybe to 2025, CAR T-cell therapy has shown incredible results with blood cancers. Obviously, Emily Whitehead is the shining example of that. I do know that there are some challenges, though, with other types of cancers, especially those that involve tumors. And I'm wondering if you can give me an understanding of where the science is in dealing with tumor-based cancers and why CAR T therapy is not quite there yet. And the hope, I guess, because I know there is hope on the horizon.
Sure. At this point in 2025, there are 7 FDA-approved CAR T cells for various kinds of blood cancer, adults and pediatrics, in the United States. And, they've really transformed the care of blood cancer. But, the common solid cancers, there are no FDA-approved CAR T cells yet. And, you can either be a pessimist or an optimist. I'm one of those who, thinks the glass is half full and we're going to solve this. So, back when we first started our CAR T cell trial in 2010, there were only 3 groups in the world doing any research with CAR T cells. And now, 2025, there's over 1,000 trials that are open. And it's not just academic groups, but it's global pharmaceutical industry, biotechnology labs. And, so there's a lot of brains now working on this of how do we make CAR T cells work in solid tumors. And I think what we've found out now is, if you don't know why something's broken, it's hard to fix it.
Right.
I think now we know why they haven't worked. And that is 2 major aspects. One is solid cancers are more heterogeneous than blood cancers. So in blood cancer, we have, in the case of leukemia, on B cells, you just need to target one antigen, and that's called CD19. That's called a lineage-specific antigen. It's on all B cells. It's on all B-cell leukemias. And that's sufficient for many, many patients. And similar for blood cancers like myeloma, where we have a lineage antigen called BCMA, B-cell maturation antigen. in solid tumors, unfortunately, that's not the case. And so it looks like we need to make and build CAR T cells to target more than one antigen at a time so that we don't have, what's called antigen escape, where we kill one dominant target on the solid tumor, but then the remaining cells that don't have that target will come out and just overgrow. So that's one problem, is the multi-targeting and issue. And there are many technologies to allow that to happen. The other issue is solid tumors are much more immune evasive, where they have ways to shut off your own immune system. And blood cancers really don't need to do that. Most blood cancers are just like a cell that's gone haywire, and it just starts dividing over and over again. And it doesn't really worry about the immune system because it can just outrun the immune system by doubling every day.
Right.
Solid cancers don't grow as fast, but they had to learn then a way to not be wiped out by your own immune system. And so now when we get CAR T cells in there, they work in our hands now, about 2 weeks in patients with brain cancer and pancreatic cancer. But then they get tired, and we call that T cell exhaustion or dysfunction. And so the field is working on ways to overcome that. Those are the two main issues: getting the CAR T cells into the solid tumor and keeping them working, and then not having the tumor escape.
Amazing. And you said there are 1,000-ish labs globally right now as we speak, working on this problem. Appreciate your optimism. I think you have to be optimistic in your field of work, don't you?
Well, and I think, Patrick, it's really important that we understand that, for many years, decades and decades, we didn't really know the cause of cancer. And then, it was really hard to say, how can you cure it if you don't know why it's broken, those cells, the cancer cells.
Right.
And now we really have lovely technology that tells us that, and we have tools that allow us to overcome that, with very precision targeted therapeutics. And so the public needs to understand the huge amount of progress. For the first time, the death rate in developed countries is going down from cancer, rather than just always go up. And then what we need to have now is continued research. And the lay public needs to understand that, they'll continue to benefit with that knowledge.
Right. I think when we first met Dr. June a few weeks ago, and we talked about the Taylor Prize, which I think is the gala's on the November 25th. Look forward to seeing you there. You said that, you're grateful for the prize, of course, but that prizes like the Taylor Prize they're an opportunity to shine a light on the concepts and the ideas of science for science sake, the belief in pure science. And, your story is clearly evidence of chasing curiosities, doing research for research's sake, chasing, again, those things that you choose to chase that can lead to results. Can you let a listener know just the value of science as an end in itself?
Yeah, I think we have so much reason now to value science because of, just you can look year after year at the Nobel Prizes. So in 2011, the first that's when we first had, as you mentioned, on August 11th, 2011, that's when our first, patient came out on the New York Times, Bill Ludwig, of being a year out, being treated with his leukemia. And during that same year, and that came as a result of over 20 years in my lab working on, the basic science of how to genetically modify T cells for maybe making CAR T cells that might actually work in leukemia. But during that same year, the number two most recognized breakthrough in science was CRISPR discovery. That ended up being the Nobel Prize that Charpentier and Jennifer Doudna shared. And so CRISPR was basically a discovery in the immune system in insects and viruses that they use so that if a bacteria gets infected with a virus called a phage, they could recognize it and be able to prevent more of them from coming in. So it was an amazing basic research that looked like it had no applicability to any human thing. Who cares about a bacteria getting infected by a virus, really?
Right.
That's a hard sell to most people in the public. But it turned out that same CRISPR system that the bacteria use to defend against a virus, that we could put that into human cells and then change the DNA in a very ordered way so that we could then make the cells more potent. And in 2020, we published a paper in Science. It was the first time where CRISPR was used in humans, and we were able to take that basic discovery and rewrite the 3 different genes on 3 different chromosomes in human T cells that we use to treat cancer patients with. And so there was an example, a very striking one of a basic science advance that can actually be incorporated right into immunotherapy.
Right.
And we see many of these things happening so we need to have basic science research where the curiosity of people can say, why is this unexpected thing happening? Maybe it will have use. And, the only way to find out is to have, research that can explore basic mechanisms of things, because if we only work on things that we already know about, we'll never know, like, that there's a CRISPR system in bacteria and that they actually have an immune system.
Right. No, I mean, I know science, and while the education, the world in general has been sort of dominated by a neoliberal idea of results and proof, and you have to get grants to then prove things. But what people really need to understand, and I think you'd agree, is that science begins with what you're talking about, the actual process of discovery and curiosity. And the scientific method is what we maybe need to sort of recalibrate, would you say? Like recalibrate our thinking politically, institutionally, to get more emphasis on that?
Yeah, I think, the United States has been a leader for over 50 years in basic science and then making medicines, for instance, and so on. And the reason that happened was we had someone named Vannevar Bush right at the end of World War II who wrote a very influential paper and a small booklet on that, with a thesis that the government should fund basic research and that will benefit the public. And then what happens is that publicly funded research can then go into the development of, for instance, antibiotics that people would use. The pharmaceutical industry could make new chemotherapy from these. So that became a very central aspect of the United States, and the way the science was judged on, because there are good ideas and bad ideas, and you don't want to chase every idea. You want to chase good ideas, and what was instituted in the United States, and it's very similar in Canada, is peer review, where you have other scientists look at ideas and proposals for research and then pick out the ones they think have the best chance of being useful science that should be pursued. And that's very different than having a political statement of what research should be done.
Right.
I think the best way to have science discovery is to have scientists propose things and then have other scientists look at that and say, is this malarkey, a bad idea, or is this a good idea? And so right now we have some danger on that in the United States on not having unfettered research driven by curiosity, but rather being politically driven on certain areas.
Right. No, it definitely seems like we're on the precipice or the cusp of something that's going to have to change to get us out of where we are now. I take comfort, and I don't know if you do, in your life's work and your life story, like we talked about earlier, some of the turmoil, the global conflicts, epidemics have often led to some of our greatest discoveries. So, here's hoping that some of the turmoil we're in now in 2025 can lead us maybe to even more discoveries when we figure things out.
Yeah, very much. I mean, right now, there is really, cancer vaccines have really never worked, but we're really on the cusp of that with new technologies such as mRNA delivery and so on. And yet, vaccines have become really something that's not being favored by our current administration. And the public needs to understand really that we really need unfettered vaccine research. It has huge potential, just as it did with the COVID vaccine.
Right. Well, that's a wonderful message. I know I'm in Canada, in London, Ontario. Just on the radio, I heard a public service announcement for getting the measles vaccine, right? So here we are. Hold tight. I hope things will improve. I think we could talk for a long time, Dr. June, and I do appreciate your time. I think on behalf of the Catalysts team and the Schulich School of Medicine and Dentistry and the Taylor Prize, a sincere thank you, not just to you, but also for your team. You're all very gracious and accommodating getting this podcast off the ground. So a big thank you to you and your team. And I wish you all the success, and I hope, you can continue with science for science sake and create more discoveries.
Well, thank you, Patrick. It's been a real pleasure to work on this podcast with you, and I look forward to seeing you in November.
Amazing. Thank you.
We hope you enjoyed this episode of The Catalysts, a medical science and discovery podcast by the Schulich School of Medicine and Dentistry at Western University, Canada, bringing you exclusive conversations with leading scientists and researchers.